Welcome back, everyone. We are in the middle of our discussion about signal transduction within a cell. We were just talking about G protein coupled receptors and how they can activate a G protein, which then can lead to the creation of cyclic AMP. Cyclic AMP can have a lot of different results in that cell, such as degrading glycogen so that you have sugar available, producing fatty acid in adipose cells. It can affect your heart rate and blood pressure in cardiovascular cells. It can affect water reabsorption in the kidney and bone resorption in bone. So G proteins are only going to be active for a very short time. They're going to need to be able to very quickly respond to changing conditions. So that stimulatory alpha G protein will become inactive. Once that happens, adenylyl cyclase will stop making cyclic AMP. And any cyclic AMP that is there is going to be quickly degraded by phosphodiesterase. So you quickly have these responses happening when the G proteins are active, and then the response ends quickly once cyclic AMP is degraded. Cyclic AMP does have a variety of different events that it is involved in, depending on what kind of cell it is involved in. But its main target in a variety of cells is protein kinase A. Protein kinase A typically exists in this multimeric form. It's got these two green catalytic components and these two reddish regulatory subunits that will be bound together and inhibit that catalytic activity of protein kinase A. But cyclic AMP can come, these little cyclic AMPs come and bind to these regulatory subunits and changes their shape. So once their shape is changed, they don't bind to the catalytic subunits so well, they're released, and the catalytic subunits are able to go off. It's a kinase, so it's going to start phosphorylating different target molecules. And that increase in cyclic AMP concentration will have different effects in different cells, ranging from bone resorption to releasing glycogen. There are different diseases that have kind of hacked this whole G protein signaling sequences. One example would be Vibrio cholerae. Vibrio cholerae is a bacteria that is found in like dirty water. You hear about it sometimes when there are floods or breakdown in city water supplies, and they'll tell you to boil your water if it could be contaminated. Vibrio cholerae causes this disease, cholera, through secreting the cholera toxin and modifies that stimulatory G protein so that it can't hydrolyze GTP, so that GTP is constantly there, constantly active. So you have this always active G protein that is going to alter this cystic fibrosis chloride transporter to alter how it is secreting salts and fluids into the intestine, which means as fluids keep being released into the intestine, your patient would have a lot of diarrhea and could become dehydrated and even die from this. Another disease would be whooping cough, which is caused by Bordetella pertussis. This is a bacteria that secretes pertussis toxin that acts on an inhibitory G protein. So that inhibitory G protein, when it's bound to the pertussis toxin, cannot inhibit adenylyl cyclase. So you accumulate fluid in the lungs, which is what causes that persistent cough. Bordetella pertussis is called whooping cough because it has just that persistent whooping. It's a very strong gasping kind of cough associated with this disease. Okay, then next we have a lot of G proteins that can act through these molecules, inositol, triphosphate, and diacylglycerol. So inositol 3-phosphate or inositol 1,4,5 triphosphate and diacylglycerol normally are bound together in the membrane as this molecule PIP2, phosphatidylinositol 4,5 bisphosphate. We're going to call it PIP2. And when phospholipase C is activated, it's going to cleave this PIP2 into diacylglycerol, and IP3. That means IP3 is now available to go float off through the cytoplasm to have effects in the cell. Your diacylglycerol can interact with different membrane-bound proteins and target signaling. Some of the signaling we get would be platelet activation in platelets, muscle contraction in smooth muscles, insulin secretion in the pancreas and endocrine cells, amylase secretion in pancreas and exocrine system, glycogen degradation in the liver, and antibody production in B lymphocytes. So when you have this signaling, the ligand binds to its membrane receptor, which activates a G protein. This G protein is called GQ. GQ activates phospholipase C. This phospholipase C is called C beta, and this is going to cleave PIP2 into diacylglycerol and IP3. This IP3 can diffuse through the cytoplasm. It can bind to something called the IP3 receptor, which is on the surface of the endoplasmic reticulum, which this is a ligand-gated calcium channel, which releases calcium into the cell. And DAG will remain on this membrane here. It is embedded in the membrane and can activate protein kinase C. Okay, so when IP3 binds to the receptor on the endoplasmic reticulum, calcium is released into the cytoplasm, which is going to cause a cell response. Calcium and diacylglycerol will activate members of the protein kinase C family, which are going to act as a kinase. They're going to start phosphorylating different target proteins, such as stimulating cell growth, regulating ion channels, changing the cytoskeleton, changing the pH of the cell, and affecting what is being secreted. Calcium ions play a very important role in regulating different cellular functions. Calcium concentrations are kept pretty low within the cell. There are calcium ATPases in the plasma membrane, which can actively transport calcium out of the plasma membrane, outside of the plasma membrane. Calcium ATPases in the ER that actively transport calcium into the endoplasmic reticulum. So calcium ion within the cytoplasm stays really low, around 0.1 micromolar. So calcium ATPases in the plasma membrane transport calcium out of the cell. They sequester calcium that's in the cell within the endoplasmic reticulum. Some cells even have sodium-calcium exchangers that further help pump calcium out of the cell. And calcium can also be transported into the mitochondria. So we have pretty low cytoplasmic calcium levels because of these pumps. However, calcium concentrations can be released by opening those calcium channels. You could open the calcium channel in the membrane. That's what happens in neuronal signaling. You could signal through G protein coupled receptors and other receptors that will activate different isoforms or different shapes of phospholipase C. You'll be activating different versions of phospholipase C. And calcium can be released from storage in the ER through an IP3 receptor channel and through a receptor called the ryanodine receptor. The IP3 receptor channel and the ryanodine receptor channels are both sensitive to calcium. So increasing calcium levels will cause those receptors to open, which further causes more calcium to be released. So this is calcium-induced calcium release because the presence of calcium opens channels that releases more calcium. Calcium release is also important in the fertilization of animal eggs. So as you have a sperm attached to the egg, you'll see this increase. This light area is showing you where calcium is increasing. So you have an increase in calcium where that sperm is attaching to the egg. And you can see it spread through the cell. So here it's still kind of at the area of the cell where the sperm bound. Then it travels more centrally and it's traveling all the way to the other terminal side of that cell. So what happens when your sperm binds is it triggers the release of calcium, which is going to release these granules and form a fertilization envelope, which blocks polyspermy. It blocks more than one sperm from binding to that cell. It also helps activate that egg, get it metabolically active and ready to start being active and dividing into an embryo. Okay, animal sperm are activated by release of calcium. Activated sperm bind to surfaces of mature eggs, triggering and releasing calcium from internal stores. And then calcium releases cortical granules to release their contents and turn this vitelline envelope into a fertilization envelope and blocks polyspermy, and also induces egg activation, which is when that egg will resume its metabolic processes, reorganize its contents so it's able to go through embryonic development. Calcium is able to bind a lot of different effector proteins. One way that it does this is with this molecule calmodulin. So this green kind of wormy shaped protein here is the calmodulin. And these 4 spots are calcium ions on it. When calmodulin is bound to calcium, it can wrap around its effector protein, so something like a phosphatase or a kinase, and activate it. Okay, so we'll come back and talk about what happens further down in our signaling in our next video. I'll see you soon. Let me know if you have any questions.